Okay, welcome everybody to the postgraduate lecture series. It's a great pleasure to introduce Ian Majewski today to you, and particularly I'd like the students to take note. So Ian is a relatively young researcher. He's a postdoc in the Cancer and Haematology division, and he, like many of you are doing now, he did his PhD at WEHI. But first he did an undergraduate at the University of Western Australia with a major in biochemistry. He did his PhD here with Doug Hilton and Vaughan Alexander, and he did a genetic screen on genes, identifying genes that are important for platelet production, but also for hematopoietic stem cell function. And then he visited the Genome Center at Washington University for a few weeks to gain experience in advanced DNA sequencing techniques, and he used that technology then as a postdoc in the Netherlands Center, Cancer Institute in René Bernard's laboratory to identify altered signaling networks in a variety of solid tumors. And in 2012, he came back to WEHI, and we are very glad that he came back to WEHI. And he's now examining genetic alterations that drive the development of cancer, but also influences the response to therapeutics. Ian received a number of fellowships, including the Cancer Council Fellowship, Victoria Fellowship, an NHNMRC Training Fellowship, and a Boomerang Award. And he published his work out of his PhD and other work in journals such as PNAS, PLOS Biology, Blood, and he also published a review in Nature Medicine. So with that, I'd like to ask Ian to come to the podium and deliver his lecture. Thank you.
I have to say, I wasn't expecting a clap at the start, but hopefully I get one at the end. So today, it's a real pleasure to come here to give you an introduction to a field that I'm really passionate about, which is cancer genomics. And what I want to do is give you an overview of some key findings in the field over the history of cancer genomics, and also introduce some of my own work in this area. So there's a lot of different ways that we can look at a genome. This is a spread of chromosomes, so containing all of the genetic information from a single human cell. For a human, we're talking about 6 times 10 to the 9 base pairs. It's a wealth of information. Contained within there is information about your hair colour, your eye colour, aspects of your personality traits, and also your predisposition to disease. So basically, we're talking about your instruction manual. And for a long time, we haven't really had the tools to read this manual. We've been restricted to looking at, say, genetic markers or looking at phenotypes and then inferring something about the DNA. But that's all changing. We're really now entering an era of personal genomics. And that means that individuals can go out there and get their genome sequenced. And one of these individuals was Steven Pinker, and he wrote an article in the New York Times about this process, about getting his genome sequenced. And I've just taken a little quote from that, where he says that DNA has long been an invisible molecule, accessible only to a white-coated priesthood, and by that he means research scientists like you and I. But today, and this holds true, for the price of a flat-screen TV, people can read their essence as a printout detailing their own A's, C's, T's, and G's. The problem is the genome is big, and it's not always easy to see what's important or clinically relevant. But fortunately, the cost of genomics is coming down tremendously, and that means we're no longer restricted to looking at one individual genome. And the reason for that drop in prices is huge changes in technology. And today's not a talk about technology. It doesn't matter essentially which technology platform you're using to read your DNA. It might involve, say, looking at millions of small fragments of DNA on a glass slide and reading out base pairs. It might involve using a pH meter to monitor addition of A's, C's, G's, and T's. It might be a bit more far out. It might have polymerases suspended at the bottom of small wells and monitoring their activity with laser light. really advanced is feeding DNA through nanopore structures, monitoring the electrophysiochemical properties of bases and reading these out. What this all really means is we used to read DNA as single bases, one base pair at a time, but we now do this with hundreds of millions of DNA fragments. That means that we no longer look at an individual's genome just in isolation. We use a wealth of genetic information that's out there. And so Eric Green gave a talk here last year where he talked about some of these resources. These include tremendous databases of genetic variants like dbSNP, the HapMap project, 1,000 Genomes, which have characterized genetic variability across really diverse populations, human populations. But it's getting much more detailed with projects like ENCODE. What you're starting to see is not just DNA variations, but also transcription factor binding sites mapped throughout the genome, real studies on the transcriptome itself, looking at different ways that genes are regulated. There's many tools that are available for viewing all of this information, for pulling it together from these diverse sources like Ensemble or the UCSC Genome Browser. But importantly for today's talk, what we're starting to see now is resources that are specifically focused on detailing those genetic changes that occur in disease, either heart and lung disease, or specifically for today's talk, a number of catalogues of mutations that occur within human cancers. So one of these is the Catalogue of Somatic Mutations in Cancer resource, which is run by the Sanger Institute, but also large international collaborations like the Cancer Genome Atlas and the ICGC. These are specifically recording all of the genetic changes that occur within cancer. So we're at a tremendous time, really. We've got a huge amount of information to bring to bear on what's going wrong with cancer, and hopefully today I'll show you that that's going to lead to new ways to treat the disease, to monitor the disease. But I still want to avoid overselling. There's some things that genomics can do and there's some things that genomics can't do. And so, as I said, lots of people are getting their genome sequenced now. We're starting to see celebrities get their genome sequenced. This is one such celebrity. This is Ozzy Osbourne. He's a rock and roll star. He was asked in 2010, after getting his genome sequenced, why did you actually do this, Ozzy? Why did you get your genome sequenced? And he talked a little bit about his wife's family history with Alzheimer's disease, but when it came down to it, he said that he really misunderstood the information, and he thought that they were going to tell him where he was going to die so that he could avoid going there. Now, I'm a big fan of genomics, but we're not quite there yet. But we need to remember that there is a limited understanding in the public about genetic information, particularly with what it can do with regards to disease. And so we need to be careful that we don't promise things that we can't deliver. The talk today is going to involve a number of things. We're going to talk about cancer as a disease of the genome. I want to give you a feeling for the fact that understanding those molecular changes can help to guide the selection of therapies for individual patients. I want to show you that genome sequencing can be used to provide new biological insights into the disease, and it can help to find new ways to treat and monitor the disease in patients. Lastly, I want to talk about how we can use genomics to improve the way that we monitor whether a patient's responding to a drug, how we can do a better job at dealing with disease progression. So let's get started. So cancer is most definitely a disease of the genome. It results from the accumulation of somatic mutations within a cell which either activate oncogenes or break down the normal control mechanisms that govern cellular differentiation, cellular proliferation. Translocations, chromosomal translocations, so the fusing of two distinct chromosomes, were the first genomic abnormalities identified in cancer, and they were identified by these two individuals, which is Peter Knoll and David Hungerford in the late 1950s. This is one of my favourite, probably my favourite photo of scientists. One, there's not that many photos that I like of scientists, but two, it speaks to that image of the white-coated priesthood, right? But two, it tells you everything you need to make this discovery. A well-annotated set of pathological slides, a microscope, a firm resolve, but it also kind of makes it look like they're levitating these chromosomes in the air with their minds. But this was a really key finding in the late 1950s. It really was a bedrock for this theory of somatic mutations driving cancer, and it actually met with a lot of resistance at the time. There were a lot of people that were still really saying that viruses what caused cancer and huge efforts going in there. We now know that they were right. We know that there's many mutations that occur within cancers and that these disrupt key cellular processes that lead to the development of a tumour. And there's a huge amount of genomics research that has gone on to define key oncogenic pathways within cancer cells. We now know, for example, that this chromosomal fusion, which is known as the Philadelphia chromosome, fusion between chromosome 9 and 22, produces an oncogenic kinase, the BCR-ABL kinase. And we know that this drives proliferation and allows these cells to maintain survival and proliferation at very high rates. There's a lot of work going on in the Institute, of course, on apoptotic regulators, and we know that fusion between chromosome 14 and 18 drives expression of BCL2 protein, and this blocks cell death. Understanding these genomic lesions means that deeply understanding the biology of these lesions means that they can become targets for therapies. We can select those pathways that are activated in the cancer cell, and we can use agents to block them. BCR-ABL itself, the Philadelphia chromosome, is really the poster child of genomics medicine. As a disease, this fusion occurs in chronic myeloid leukemia, and chronic myeloid leukemia was defined as a disease in the 1840s. As I said, we had the identification of this recurrent chromosomal alteration. Also particularly important here was the work of Janet Rowley in identifying the fusion between 9 and 22. But later we have the advent of molecular biology, the cloning of the BCR-ABL gene, and the use of a specific inhibitor then as a therapy for CML. And this has had tremendous success really in the management of one of these horrible diseases. And at the time this caused really great hope that we'd be able to develop a drug for every distinct type of cancer. And there has been a lot of progress. Understanding the pathways that are altered within cancers has led to the development of a number of different targeted therapies. And I've just shown a few examples here. So of course we've mentioned BCR-ABL fusion being treated with a small molecule inhibitor, imatinib. Later on I'm going to talk quite a bit about fusions in lung cancer, and EML4-ALK is another fusion involving a kinase which can be treated with the small molecule inhibitor crizotinib. We also have antibodies that can be used as therapies. So amplification of growth factor receptors like HER2 or EGFR can also be targeted with antibodies. And I've mentioned this mutation in BRAF, an activating mutation in BRAF, which is particularly frequent in melanoma. This is a wonderful success story because it utilises a drug that's been designed specifically to hit the mutant form of the protein and not the wild type. And that hopefully leads to less effects on normal tissue. It's not only important to know which pathway has become activated in a tumour, it's important to know at what point in the pathway it's become activated. And I want to use the example of EGFR-based therapies in this instance. What we have here is just a schematic diagram of the EGFR pathway. Like many of these signalling pathways, it starts with a receptor tyrosine kinase, which becomes activated and then through a series of effector molecules leads to a change in gene expression and encourages proliferation or survival. The therapies themselves, either the antibodies or small molecules that attack the kinase domain of EGFR, act at the level of the receptor. What this means is if we have a mutation that's downstream of the receptor, we're not going to see therapeutic benefit. The signaling is still going to be active in this pathway, it's not going to respond to therapy, and the patient's not going to see any benefit from what is a relatively expensive drug. And I like to think of this in terms of an old Chinese saying, which says, if you want to be successful in battle, then you really need to know yourself, but you really need to concentrate on understanding your enemy. And this is the kind of thinking that really led to the first cancer genome studies. Really an effort to understand every alteration that's occurred within the genome to explain the biology of a tumour. As Anna said, I was fortunate enough to visit Tim Laid's laboratory in St. Louis just as they completed this first cancer genome study. It was a myeloid leukemia. The group's mainly focused on myeloid leukemia biology, and it was a major technical achievement at the time. the coverage was actually relatively low, so they produced 30-fold coverage of the cancer genome and 30-fold coverage of matched skin samples. And they used this information then to identify somatic mutations within the tumour. I want to go back to the primary data from this paper, and I want to illustrate 3 points which I think are applicable to any type of cancer genomic work. The first point I want to make is coverage is important. So this is a list of genes in the black that were found as mutated in the first AML genome. Now the coverage was relatively low, right? 30-fold on average across the genome. That means there's bits of the genome that you're going to have in higher than 30-fold, bits that are lower, and therefore you might not see every mutation. That's why you're starting to see other studies like exome sequencing where the coverage is much higher just over the coding regions of genes. When they went back later, in later studies, they identified an additional mutation in this sample, which was in the DNA methyltransferase 3A gene. The second point I want to make, coverage is important, is point number one. The second point is cancer genomic studies have major potential to identify new genes that are involved in disease that have a major role in the development of the disease and also disease biology. So, two successes that have come out of Tim Lay's work are the identification of mutations in DNA methyltransferase 3A and also in the metabolic enzymes IDH1 and IDH2. These are particularly important because if we actually look at cohorts of patients that carry mutations in DNA methyltransferase 3A, and that's shown by these bars down below, they have a much worse outcome than patients that do not carry mutations in this gene. That means that the traditional therapies that are being applied in myeloid leukemia do not work effectively for patients that carry mutations in this gene. This is an important finding because it really says there's something different about the biology of this disease, and potentially we should be looking at new ways to target this disease specifically. The third point I want to make, and this is, as I said, applicable to any genome sequencing effort, is about the diversity that we see within cancers, the diverse range of mutations that we see. So here, we've got the list of genes that's mutated, some information about the exact type of mutation, the actual primary coverage, the number of reads, so you can see that it was very low. And in the last column, what I've added is the recurrence rate. So if you take 180 additional AML samples, do you see mutations in that gene? And for the vast majority of these genes, this is the only patient where we see a mutation in that gene. For 3 of them, for 2, they were very well-known players, and for DNA methyltransferase, they found a new gene that was then mutated in quite a significant population of patients. But the diversity is the scariest thing to come out of the first cancer genome studies. This is something that we've known about for quite a long time. So even with the first studies, these were performed by Bert Vogelstein's group, published in 2007, where they sequenced all the coding regions of a small number of tumours. They interpreted their results as that the cancer landscape itself is one of mountains and of molehills. What do I mean by that? This is a 3D plot. They were very popular in 2007, I think, but it's not very complicated. It's the first gene on chromosome 1 and then going on and listing them all the way to the end of the last chromosome. So it's just putting the genes in order and spatially separating them to give you a bit of, I don't know exactly. But what they've done then is taken a small handful of colorectal cancers and plotted the mutation frequency for each one of these genes. And what you can see is real standout peaks for these genes, APC, KRAS, P53, PI3 kinase. So these genes are frequently altered in cancer. They're the mountains for colorectal cancer. But there's many genes that show low mutation frequency. That's not to say that they're not important. So how do we go about interpreting these genes that have low mutation frequency? There's 3 real reasons that we see some genes that show low mutation frequency. The first is the disease is inherently driven by the random accumulation of mutations. So some genes are going to carry mutations and they're not going to be functionally important at all. These are sometimes referred to as passenger mutations because they're just along for the ride. But there's two other deeper reasons about low mutation rates. The first is, if we're just considering mutations, so changes in the DNA sequence, that's only one way to alter a gene function. There's many ways to alter a gene's function. You could look at copy number changes, so amplification of the gene. You could look at methylation of that gene, altered targeting of microRNAs. There's many different ways to alter a gene. So mutation is only one way of looking at it. The third reason, and this is also particularly important, is we need to think about pathways, not just individual genes. And so what I've shown here is a pathway diagram. This is one of the first Cancer Genome Atlas publications, which is a study of lung cancer. We've talked already about the importance of receptors driving cellular growth and the network of genes downstream of the receptors. Now, if we consider one component of this big signalling network, like AKT, we can see here that the mutation frequency is only 2%. It doesn't seem very important, right? But if we consider the broader network around AKT, then we see a much larger proportion of patients that show activation of the PI3 kinase pathway, receptor tyrosine kinase signaling. And this means that potentially these patients can be grouped together and treated with similar therapeutic agents. So the diversity has actually had major implications, and it's had implications for how we design genome-wide studies. So now you see big international cooperatives, the TCGA project or the ICGC project. And to deal with this diversity, both in the type of mutations that you see and in the pathway level, they apply many different types of technology. So they'll perform exome sequencing, which is targeted sequencing just of the coding regions for the genome. They'll do gene expression profiling, copy number analysis, methylation analysis, microRNA profiling to try and get at every distinct mechanism that can alter gene function. The other thing is the numbers are getting bigger. So now what you'll see for a standard study is a study of maybe 300 to 500 tumours, and that's to deal with this great diversity in the disease. There's been large cooperative efforts on many different types of cancer, and these have been tremendously successful. I want to highlight a few individual examples, like the identification of ARID1A mutations in more than 50% of clear cell ovarian carcinomas. So this is the identification of a new tumour suppressor that's mutated in over half of this one disease type. Other successes, EZH2, the polycomb group protein, has hypomorphic mutations that are present in diffuse large B-cell lymphomas. And in a similar vein, this is not particularly part of TCGA, but mutations in histone tails that also change the epigenetic regulation of these proteins found in brain cancers. So these are examples where you see quite a high mutation frequencies. These are mountains. But if we just consider mutation frequency itself, if we look at serous ovarian carcinoma, here they sequenced 316 patients, found 20,000 mutations, but really, at the level of mutations, only relatively few genes showed recurrent mutations, p53 being the standout. So no real new insight just by looking at mutations. If we take the serous ovarian carcinoma as an example though, starting to integrate this information using all of the available tools, we actually start to see significant enrichment in particular pathways around DNA repair that are altered in a much higher proportion of patients. So data integration has a tremendous ability to start identifying these important pathways within cancer, and that's only going to get better as we expand into other fields like epigenetic regulation, better understanding of metabolomics, and also real measurements of signaling pathways within cancer cells. So I hope now that I've given you a feel of really the current state of the field. We're making lots of different measurements, and there's genomic information out there for several thousand cancers. I want to now concentrate on a few examples of how I think having this information is going to change the way that we treat and monitor the disease. So first of all, I think it's going to be a really good tool in respect to understanding why some patients respond to a particular drug and why some people fail on therapy. And this is a resource that I want to highlight that was published both by the Broad and by the Sanger Institute. what they've done is actually take very large panels of cancer cell lines. They've done thorough genomic characterization. And there's a lot of questions that can be asked about the relevance of using cancer cell lines, but there's a lot of things you can do with cell lines that you can't do with primary patients. So they've taken hundreds of cell lines and treated them with thousands of clinical agents, and then looked for the types of mutations that would either govern sensitization or resistance to a therapy. We've talked already about the EGFR pathway and EGFR-based therapies in cancer, so I've highlighted this one again. And what we've started to do is actually take unbiased genomic information and look for mutations that drive resistance to EGFR-based therapy. So here we've got a plot indicating resistance, and here a measure of statistical significance. I said before we knew that mutations in KRAS cause resistance to EGFR treatment, and indeed they found KRAS as a predictor of resistance. They also found other pathway components like BRAF and other regulators of the PI3 kinase pathway. Now these are relatively simple approaches and testing for simple relationships, but these exact kind of tools are now being brought into clinical trials. So instead of doing the clinical trial and then thinking about resistance, you're starting to see people testing up front for genomic changes that might be driving resistance. The other key area is really in disease detection and monitoring therapy response. Now, I talked before about the Philadelphia chromosome, and that was discovered in the late 1950s, early 1960s. Even at that time then, people started to think, can I use this chromosomal alteration to monitor disease? And they started doing that by doing chromosome spreads and tracking this chromosomal alteration and using that as a surrogate to track disease load. Now there's much more fancy tools that can be used to monitor BCR-ABL measure, quantitative PCR measures, but this same type of approach can actually be extended to any type of genetic alteration seen within a disease. So although these translocations, recurrent translocations, are less common in solid cancer, we can perform a detailed characterization of the genome of a tumour and then design specific tools to monitor those genetic alterations in a patient. And so I'm going to go into this in a little bit of detail because it's important also later in the talk. This is work from Ulton McDermott, but also similar studies were going on at the same time at Bert Vogelstein's group at Johns Hopkins. What they did was they started with a cancer patient. They did a detailed genomic characterization of the tumour. They identified particular rearrangements in the cancer, and they designed specific primers to be able to amplify those alterations. Now, what they then did was actually extracted serum from the patients. Now normally, serum is the cell-free fraction of blood. Normally there's not a lot of DNA in there. But when you have a solid tumour growing, what you find is that there's a lot of necrosis, a lot of death as the tumour expands, and small amounts of DNA actually make their way into the serum. So we can extract serum DNA and then test for the presence of this particular alteration and use that to monitor the load of the tumour within the patient. And this is an example of just that. This is a patient at presentation with disease. The patient has high levels of this marker within their serum DNA. They have surgery and therapy and this drops away. But about 2 years later, we start to see this marker come up again in the blood. And importantly, that's before any obvious signs that disease has relapsed. So potentially this is a non-invasive way and a cheap way of identifying disease that's coming back rather than doing an MRI scan at 3 months. You can also extend this to try and assess whether a therapy is working by injecting patients with therapy and seeing whether that's causing a measurable drop in the tumour load. This is a second example that I'm really excited about, and it was exciting enough that they also wrote an article about this one in the New York Times. This is some work from Bert Vogelstein's group again, and what they did here was really clever. They used the knowledge of the genomics of particular diseases, endometrial cancer and ovarian cancer, and they used this to develop new screening techniques. So they did a detailed characterization of endometrial cancer and ovarian cancer, identified the genes that showed the highest mutation rates, and that is these 12 genes shown here. They then went to material from pap smears. They extracted the DNA from those pap smear samples, and they used very sensitive techniques to detect any types of mutations in the pap smear material. And that could be used as a screening tool to detect early stage tumours in women with endometrial cancers. So I really like this example because it uses the knowledge of genomics, but it also capitalises on existing structures within the healthcare system. So you're not really rocking the boat too much if you want to do this type of test. I do want to add though, there's a lot of work going on now to see whether this is really a realistic approach that's going to provide a lot of benefit or not. And a lot of work has to be done here, but I think it's very exciting. So now I want to introduce some of my own work. I said before that these gene fusion events, like BCR-ABL and also EML4-ALK, are important driving events in the development of some tumours. And I want to look at the way that we do current testing to detect these translocation events in a solid cancer. And so for EML4-ALK, this is done with a split fish assay, and I'll tell you what that is. Split fish is where you're interested in one particular gene, the kinase ALK in this instance, and you're interested in rearrangements of that locus. So what you're going to do is have two probes, one that hybridizes to the 5' end of the gene, shown in red, and one at the 3' end of the gene, shown in green. In a normal cell then, these probes should be together, and you should have, if you look at the nucleus, 2 yellow spots corresponding to the 2 normal copies of ALK. With a rearrangement, however, these probes are going to be moved apart, and what you're going to start to see is individual spots of green and red. And I've shown an example of an ALK translocation sample here, I believe, where if we take a close-up view of the nucleus, what you see is 1 yellow spot and 1 red and 1 green spot. So these 2 probes have been separated. This is a fantastically successful and sensitive assay. It's been developed as FDA-approved diagnostics, etc. But we were thinking in our group, together with René Bernards and Lorenza Mitberger, why not use genomic tools to test any type of translocation events in all kinases at one time? These types of assays, they only look at one gene. Why not look at all of the kinases? And so to do that, we developed a capture enrichment technology to focus our efforts on understanding whether we could see any alterations in kinases in a particular cancer. And the idea goes like this. What we're looking for is these transcripts where a kinase gene has been fused with another gene and is might be activating it, driving high expression, etc. So what we did was we built sequencing libraries from these samples, and then we used biotinylated capture probes to actually pull out the kinase transcripts, because those are the ones that we really wanted to sequence. Now, in my other work, I do a lot of sequencing on DNA samples, and this is really wonderful if you're interested in identifying mutations, individual mutations in the sequence of a gene. So, for example, what I'm showing here is a schematic of the genome with exons that we're interested in. Those that we've captured will get lots of sequence reads over that exon, and we'll do a good job at identifying point mutations. The problem is things like translocations typically occur in intronic space. They occur away from the exons. And that means with this type of technology, we've got limited potential to see this event. In the case of cDNA though, so produced from the RNA from the sample, the splicing machinery has done the work here. These exons are right next to each other. And by enriching for the kinase portion of the transcript, we'll get a lot of evidence then for this fusion event. So this is what the result kind of looks like. Here we're comparing the coverage of each individual gene after we've done the capture or before we've done the capture. Each spot here represents a gene, and the spots that are in pink are those that we've performed part of our capture library, the kinases. So what we see is a dramatic boost in the coverage at this site, about a 20-fold boost in the coverage. This means that we can do a sensitive job at detecting mutations and structural alterations in these genes. This worked really well in some pilot studies, so we expanded the study then to look at 96 non-small cell lung cancers. So these are solid tumours, And within non-small cell lung cancer, there's a couple of histological types. There's squamous cell carcinomas and adenocarcinomas, and that becomes important as we go on. So we built mRNA libraries from these 96 samples. We captured out the kinase genes, and we performed sequencing on a high-seq machine. This was paired-end sequencing, which allows us to read both ends of small cDNA fragments, and that's important because we might want to look at where they're located at the genome and get proof that they might be located further away than they should be. We sequenced 6 samples per lane, so for those that are interested in the detail, that's about 20 to 30 million reads per sample, so not that much. We then went on to detect any potential fusion transcripts, and we did this with a couple of different methods, but I'm really happy with the collaboration we developed with the Murdoch Research Institute, and I'm going to show some results from Nadia Davidson there and Alicia Oshlag, and that's been a really, really nice collaboration. So we're looking for 2 transcripts that are stuck together, and we found a number of these things. And what I'm showing is just an example of one such fusion transcript, and this is belonging to a known fusion between EML4 and ALK. Here we can see the transcript itself, the portion that belongs to the EML4 gene in grey, the portion to ALK in blue, and the yellow portion which contains the region where we're seeing the fusion between the 2 transcripts. Underneath, I've just shown some sequence read, paired sequence reads, and the yellow region is important because it's that bit that gives us the evidence that these 2 genes are fused together. We can also see here that the coverage is much higher at the ALK portion of the transcript, and that's because that's where our capture probes are that are pulling down that individual gene. We identified many different potential fusion candidates, and I've just shown a few of them here. These are in-frame fusion proteins, so likely to be changing the functional activity of that particular kinase. The first one we identified, I've talked about, EML4 ALK, was already known, but we also identified a novel fusion partner for ALK, and this was the STRN gene. So we went on to validate this fusion event between STRN and ALK, and we did the same thing. We used a split fish assay to prove that the locus was rearranged. So we have the 2 probes again, and for this individual sample where we detected the fusion, if we look at the nuclei, what we see is 2 red signals and 1 yellow. So the yellow corresponds to that normal unrearranged locus, and the 2 red spots represent the ALK locus that has been rearranged and then amplified, so we have the 2 signals. This has the same consequence as many different ALK fusions. It boosts the expression of the protein, and this is immunohistochemistry looking at expression of the ALK protein in that tumour sample and showing that it's very high. And this means that this patient would probably be a good one to apply these small molecule inhibitors of ALK kinase. The next fusion I want to talk about is this one between FGFR3, which is the fibroblast growth factor receptor kinase 3, and TAC3. And here we had a very large amount of the kinase involved in the fusion and a small amount of the TAC3 gene. And if you look at the domains present in the fusion, we see that it maintains almost the entirety of the FGFR3 gene, including the tyrosine kinase domain. And all we get at the end from TAC3 is this one TAC domain, and this is involved in dimerization, so potentially bringing the 2 kinase molecules together. If we look in the samples, all of the samples that we sequence, we can look at the expression level of these 2 genes. And we see that the one that has the fusion has very high level expression of FGFR3. The nice thing about having very deep coverage of the kinases in all of these samples, though, was we could also go back then and look for mutations that occurred in FGFR3 in any of the other lung cancer patients. And we found 2 additional activating mutations in FGFR3, and that's shown here. So FGFR3 has a number of hotspots. These are frequently activated sites. This is a very highly mutated gene in bladder cancer. And we found one of these mutations, serine changing to cysteine at position 249. Here I've shown a sequence coverage histogram from the next gen result. So the C is changing to a G. And underneath, we've also gone on to confirm this using traditional capillary sequencing. So we both have, using this one assay, activating mutations through a fusion event and activating mutations. We also wanted to get a feel then for how important FGFR3 was in the development of these different types of lung cancer. And so we next applied a simple tool called a tissue microarray, where we had small samples out of many different tumours, both squamous cell cancers and also adenocarcinomas. And we found that about 7% of squamous cell carcinomas express very high levels of FGFR3. Importantly, when we took these 10 samples that showed very high expression of FGFR3, we could find 2 additional cases where FGFR3 was fused to TAC3, so enriched for this fusion event. We didn't detect any expression of FGFR3 in adenocarcinomas, and that tells us this is probably something specific for squamous type disease. The really exciting thing, and the reason that we're really interested in the presence of this FGFR3 TAC3 fusion, is that this event's also been described in glioblastoma and in some bladder cancers. And that means we can kind of hope to deliver on the promise that an oncogenic lesion like FGFR3 will be a good target for therapy. It's found in many different disease types. It has strong oncogenic activity. And importantly, in models where we drive transformation by FGFR3, this is work that was published last year in Science, those tumours respond very well to small molecule drugs that target the kinase. So potentially, we would like to see application of FGFR3 inhibitors within squamous disease, and I think this is possible. Right now, FGFR inhibitors are being trialled in squamous cell lung cancer, specifically with patients that show amplification of the FGFR1 gene, so a related receptor. But we think that it would be good to broaden the criteria of these clinical trials to include patients that have either activating mutations or this translocation event. You've found a driving oncogene within your tumour. You've found a small molecule drug that's going to inhibit that oncogene. The problem is that that's not the end of the story. The landscape is dynamic, and by that I mean the tumour is going to continue to accumulate genetic alterations. This results in heterogeneity within a disease, and what this means is that therapy is often a selective pressure. We're going to need to think about how these cells evolve in response to therapy, and Peter Knoll was talking about this in the 1970s, about heterogeneity within a tumour. These kind of studies have been confirmed and looked at now at the single cell level. So this is a study from Cold Spring Harbor where they took out individual nuclei from a tumour, sequenced the genome, and could prove this hierarchical relationship within the disease. We need to think about how tumour cells evolve. Targeted therapies can produce spectacular results. They also produce lasting results for some patients, but for some, the results, the effect doesn't last. This is some evidence of the dramatic influence of small molecule inhibitors. This is a melanoma patient treated with a BRAF inhibitor. And I believe some of this work was done at the Peter Mac together with the team there working on BRAF inhibitors, Grant MacArthur. The problem is often we see a relapse, and a common mechanism of resistance is actually a secondary mutation in the target itself. And so, for example, BCR-ABL, it's been known for a long time now that some patients will progress on the therapy, and often they'll accumulate mutations in the kinase itself that will stop the drug from binding effectively in that pocket. And these types of mutations in the target are commonly seen with BCR-ABL, ALK, and also the EGFR receptor. Now, I should say there's now also the development of secondary inhibitors that will deal with these mutant forms of the targets. But sometimes the resistance mechanism is even more complex. So if we actually look at these BRAF inhibitors, just in the last couple of years, many different resistance mechanisms have been defined, whether that's mutations of components of the pathway, upregulation of different growth factor receptors, loss of tumour suppressors, upregulations of other kinases, many different distinct alterations. And so that's why it makes sense in these contexts really to take an approach that looks at all potential mutations that might be occurring. Now, I wanted to give an up-to-date review, so I'm quite glad this paper came out 2 weeks ago in Nature. It's from a group in Cambridge, and they're working very much on this idea of using serum DNA as a marker of the tumour. What they're doing is taking DNA from the serum and performing exome sequencing, sequencing all coding regions of the genes. And what they're then looking for is actually mutations that are enriched as a patient is treated with a drug. So what they're looking for is these mutations that are becoming more prevalent within the tumour and that are therefore candidates for driving resistance to that disease. This all sounds a little bit scary. I don't want to leave you with a feeling that it's all hopeless. The fact is, as these cells evolve and change, as we see changes in the tumour, changes in the signalling pathways within the tumour, those differences then can become targets for therapy, and we can start to see strategies that take advantage of the changes occurring within cancer cells, specifically to kill them. Now, I want to finish briefly with 2 areas that are showing rapid development in terms of cancer biology, and then lastly finish with a couple of thoughts about my views on cancer genomics studies. So 2 growth areas. One is in epigenetics. We know that DNA exists in a complex protein-rich environment, which is highly dynamic. These modifications to DNA and proteins occur and change very quickly. We know that the epigenetic environment is important because we see mutations in many different epigenetic regulators across a diverse range of cancers. And so understanding these changes and targeting these modifiers is going to be critically important. Even further away, host-environment interactions are also going to be important. So we've started to see the NIH investing in understanding changes in microbial populations within the body. And this is also investigating links between the microbiota and diseases, including cancer. But more broadly, sequencing can help to reveal environmental factors like viruses or the evidence of particular carcinogens that are causing cancer to develop in the first place. So lastly, I want to finish with my thoughts on how we can use genetic information in cancer. For me, genetic sequencing ultimately results in a list of A's, C's, G's and T's, right? We talked about that at the start. It's in the interpretation of this information that we have strength. And so the actual genome sequencing itself, for me, is kind of like an X-ray. It gives you some insight into the underlying structure within the disease. And for some of us, for most of us, we can probably identify some things that look particularly nasty in this picture, right? So let's imagine that the fangs themselves might be BRAF mutations or P53. We have some idea of things that we might like to avoid. It's in the extra detail though. It's in the transcriptomics, understanding epigenetic regulation, metabolites, phosphoepitopes, that we actually start to get some colour. So maybe this gives us a slightly different picture of the disease itself. The critical thing though, understanding which of these is the dangerous one, comes down to having clinical experience. So understanding the mutations that occur within a disease and which ones of those are associated with a poor clinical outcome and that need to be targeted. So that's pretty much the end of my talk. I hope today I've given you a feel for the way that we can use cancer genomics information both to understand why cancer happens, to monitor that disease within patients, to understand which therapies are going to be best applied to treat the disease. And I think really if we apply these genomics technologies, we'll be in a much better way to handle the disease and ultimately have better results for patients. And so there's many people that are involved in the work, and the ones highlighted in blue have played a particularly important role into the work, my work that I've talked about today. And with that, I'm very happy to answer any questions that you have.
So thank you, Ian, for a very clear presentation. We want especially questions from students, but not exclusively. Please. Yes.
You mentioned the heterogeneity within the tumour population and now the ability to sequence far deeper into the genome of the cancer population. Do you see in the future using a diagnosis, say a deep sequencing approach to understand not just the bulk majority of the tumour cell population, but also the minor subclones that might be more prone to relapse, and then from that taking that to a multi-pronged attack at the cancer from the outset as opposed to treating with the normal regimen?
Yeah, absolutely. I mean, the allele, the prevalence of a mutation really tells you a lot about the importance at that particular stage in the disease, and monitoring that dynamically over the course of a treatment tells you then about the presence of those clones, about resistance mechanisms that might be operating. Once you perform these studies on many hundreds of patients, actually understanding how a drug works, then you're going to understand very well about the escape routes from a particular therapy. So
take a preemptive approach to where it might go from there?
Well, both. I think right now we're still at the stage where you need more information about where the resistance, what are the resistance mechanisms, and maybe it's even better to have more sensitive techniques rather than full genome sequencing to monitor those specifically. But that kind of path leads you towards better combination therapies that block off those resistance mechanisms before it gets there.
You take this question and then.
So with the preemptive strike sort of method, that we after a while we know, okay, it's going to mutate this way if we treat it with that drug, and you do preemptively treat with a new drug to block that, wouldn't they just keep mutating a different way and we're creating new and new ways and sort of very deeper?
Absolutely, that's possible. But I think what I hope that I've shown you is that also there's great progress in detecting disease at an earlier stage, right? So the best therapy is if you can actually remove a tumour with surgery at an early stage in disease where it hasn't spread at all, and with appropriate chemotherapy get on top of any type of metastatic disease. So these tools are going to be just as important in finding that disease as soon as possible, and think about expanding those the approach that I talked about with pap smear screening. Think about those kind of techniques expanding into everyday life to do a better job at finding those people that have cancer before it's at an advanced stage.
So I was interested in the serum test for recurrence of DNA from tumors, and you characterized it as saying that at the site of recurrence there's some necroses and cell damage and DNA escapes and is in the serum. I was just wondering, when people do that, how advanced is the local recurrence for material to get, detectable material to get into the serum, and is it a better thing than looking, as you said, like MRIs or something like that? Is it maximum?
Absolutely, that needs to be worked out now. So there's a lot of people that have, previously there was a lot of focus on actual circulating tumour cells and using those as a source, and that potentially you can get at a very early stage of spread. I'm not an expert on the stage that a disease needs to be at to produce serum DNA in the serum, but I am aware that there are screening efforts going on now currently, tests to see whether this can detect, which stage this can detect that. And that's particularly active in fields like ovarian cancer, where they do such a poor job. It's so difficult to find disease at an early stage where this kind of test would have a major benefit. So it's a very quickly developing field, but I think it's got a lot of potential.
You talked a lot about detecting a lot of these sort of emerging mutations and so forth, and things that are causing disease to progress or grow on. Are you expecting there to be a lot of commonality in what emerges? I'm just sort of thinking about how we deal with the spectrum of what turns up and how we then treat it. I mean, it's one thing to detect all of these progressive mutations that are occurring, but how do we treat each of them? Are they going to be unique enough that we can personalise things, or are they going to be common enough that we can develop individual tools that can be used across multiple people? How do we deal with this amount of mutational information that we have?
Absolutely. I think right now that might depend on the particular type of drug, but really it's a broader question. for example, all those diverse mechanisms that produce resistance to BRAF inhibitors, they all result in elevation of ERK activity. That's the key pathway that's supported the tumour's growth during its development, and that's where it's going back to. But you could see the situation where there's going to be many diverse mechanisms, say involving drug transporter pumps, all of these. You can think of hundreds of different ways potentially for this to happen. The question is, which are the most prevalent mechanisms? And exactly right, which of those are common, act in the same way, and then can be targeted with one therapeutic agent? So it's always a numbers game, but I think those are particularly important studies. And at least if you've done the genomics, you've got the list of genes, you can do the functional test to see which ones are related and which ones have different activity.
I was wondering if about passenger mutations, so functionally relevant mutations occur frequently, but there was also a consideration that you could have mutation hotspots, and the number of studies that have done now should reveal whether you can have mutations, frequent mutations that are functionally irrelevant.
Yes, you can. So there's particular sequence contexts that are more prone to deletions, rearrangements, and that's known now. And so what you would often see in a study is people starting to compare mutation rates, not only cancer versus normal, but also then start to look across cancers, and you see a small set of genes that either through they're closer to telomeres or centromeres, some processes causing them to have slightly higher mutation rates. Also genes that are completely functionally not important anymore might accumulate mutations faster.